Ashok, it was a really lovely talk you gave.
Thank you, Gordon.
It's 35 years, I think, since I joined the rank of cancer consultants, and mitotic spindles were what I taught medical students about, and then they got lost. So now we're back to how the cells divide and so. So what's brought it back? Has this been technology, or is it a fad, or what?
I mean, that's a really interesting question. I think there are actually several reasons behind this revival and interest in mitosis and the process of chromosome segregation. I think the first thing actually goes to the arena of therapeutics. As you're probably aware, drugs like Taxol have been in use now for decades, and they are amongst the best cancer agents, anti-cancer agents that we have. But it's still a problem that we can't identify those patients who are most likely to benefit from taxanes with tumors at different sites. So one interest, I think, that has sparked a revival of thinking about mitotic spindles is simply, here you have these really successful anti-cancer agents. Clinical resistance is still a problem. Tailoring it to the right patient is still a problem. Can you understand these problems, overcome these problems by delving deeper into the biology of the mitotic spindles? And I think the answer has been yes, you can. I think the second thing is our growing realization over the last few years that cancer cells, as they grow or evolve, I should say, essentially acquire chromosomal instability. They become aneuploid, and this is true of almost all of the common epithelial tumors that we would most like to treat. These mechanisms clearly impinge on the way that these cells are going to respond to existing agents like taxanes or even many of the new targeted agents in development. And I think that is really the final reason, which is there's been a lot of drug company interest over the past few decades in taxane workalikes because taxanes have been so successful. And I think it's a combination of all these factors that have revived interest in the mitotic spindle.
Do you think it's going to solve some of the problems we've been seeing on the failure, relatively, of the kinase targeted molecules? I mean, there's been a massive amount of investment by big pharma, but if you look at the targeted therapies, the last 3 years, each year, the FDA has approved less and less and less of these drugs. So Plan A is failing. So is this Plan B?
It's one facet of Plan B, because I think you've put your finger on a very critical issue, which is that a lot of anti-cancer agents are failing in trials of clinical efficacy. And this is a very expensive stage to fail because you've already invested hundreds of millions of dollars into getting these drugs, and then you find that you can't show that they actually work in patients. So what are the challenges underneath that? Personally, I think there are two major challenges. Challenge number one actually relates to the types of targets, the number of targets that we can drug. If you look at the human genome, it encodes perhaps 30,000 or so proteins. The best current estimates are that we can drug less than 10% of those targets. And a large number of the so-called druggable targets are indeed, as you point out, enzymes like kinases, and the inhibitors are ATP site competitive inhibitors. These are great, they do work, they do have the right effects, but they also carry with them some inherent problems. Problem number one is that the structures of many of these active kinase domains are similar between families and even within superfamilies. So most of the kinase inhibitors that are in development are not highly selective. The second thing is, because you're attempting to inhibit a very fast turning over enzymatic reaction, you need very high potency, and that sets a major challenge to getting through to a good clinical candidate. The final problem is one of therapeutic index, which is essentially the minimum dose that you need for efficacy compared to the maximum tolerable dose. Because of the problems with selectivity, with off-target effects, because you need such a high potency to inhibit enzymes like kinases, the therapeutic index of some of these compounds is perhaps not optimal. And for all these reasons, or a combination of all these reasons, I think we need to look elsewhere. I think one place to begin to look is to move away from simple ATP competitive inhibitors of enzymes and begin to look into the macromolecular interactions that regulate enzymes. So although all enzymes like kinases turn over ATP and you can inhibit by ATP, nonetheless they're also regulated by interactions with other proteins. These protein-protein interactions may activate enzymes, may inhibit enzymes, may bring substrates to enzymes. And if you are able to target with small molecules these protein-protein interactions that regulate enzymes, then you have the potential to develop more selective agents.
But that's not easy.
It's not easy, and at the moment we don't have any technological platforms that will enable us to systematically drug protein-protein interactions that regulate enzymes in the same way as people do very routinely now in industry for drugging kinases. And in fact, this is a focus of a Cambridge-wide initiative that I've helped to coordinate, the so-called Cambridge Molecular Therapeutics Program. One of our aims over the next 3 years is to combine the strengths of Cambridge science in structural biology, in synthetic chemistry, in cancer biology, to see if we can develop a toolkit for attempting to drug some of these protein-protein or other macromolecular interactions. It's very much an academic effort which aims to explore platforms and tools. It doesn't aim to make clinically viable candidates, but what we hope is that by perhaps providing such a toolkit, we may make it possible or even easier for other people to go after interactions like that more systematically.
Chemists needed?
Definitely. Chemistry is essential if you are going to develop tools. But I think the other aspect is you really need to bring biologists together with people who have chemical or physical sciences knowledge, because when you start to attack problems like this, there isn't much precedent. So for example, even simple questions like how potent should disruptors of protein-protein interactions be in order to achieve the desired therapeutic effects? We don't know the answers to even simple questions like that. And I think it is really going to take a transdisciplinary effort pulling together people in order to make this.
And how would you know it worked? Have you got ready ideas for biomarker or for
yes, I mean, I think
measurable things which would help you get the answer to those questions quickly in the early trials in the clinic?
Yes, indeed. I think we would be going in our initial cellular studies for effects on phenotypes. We're also developing imaging readouts which will help us to understand whether or not these prototypic inhibitors are in fact selective or working at the target. In the clinic, there is undoubtedly a need to develop biomarkers. And in fact, a few minutes ago, I alluded to two major challenges, not one. I've been talking only about the one, which is targeting new types of interactions for making drugs. The second is actually, how do you introduce drugs into the clinic? And this is a problem even for well-validated compounds like the kinase inhibitors, the classical kinase inhibitors we were talking about a few moments ago. And I think your point about trying to understand what the biomarkers are that will tell us whether these compounds are working, biomarkers which will identify those patient groups in whom they're most likely to be effective, these are critical. And I foresee that trial design will become not, if you will, a guessing game, but perhaps a game in which you can select those patient populations most likely to benefit from the therapy. And this will, I hope, in time lead to quicker and more efficient ways to show clinical efficacy.
Thank you very much. I wish you very, very best of luck.
Thank you very much, Gordon. It was nice talking to you.